Tonight's lecture concerns animal diversification. And here are our learning objectives for this chapter. We will be able to define an animal and the four key distinctions that divide the animals. We'll be able to divide and describe the invertebrates, including sponges, cnidarians, flatworms, mollusks, arthropods, and echinoderms. We will also be able to define and describe vertebrates, including amphibians, birds, and mammals, and we will discuss human evolution. Looking at a kangaroo, it's obvious that it is an animal. The same goes for a mosquito, squid, or earthworm. Things get a bit more difficult, though, when you look at a sponge. Is it an animal? Is it a plant? Is it something else? The sponge is classified as an animal because it shares certain features common to all animals. Surprisingly, considering the enormous differences among even the few animals mentioned here, just 3 characteristics are generally sufficient to define an animal. 1. All animals eat other organisms. In contrast, plants and some bacteria manufacture their own food through photosynthesis. 2. Animals move, at least at some stage in their life cycle. That qualifying phrase is important because some animals, such as mussels and barnacles, move only during a period called the larval stage that comes early in their lives. When they become adults, they are sessile, fastening themselves to a surface and staying there. Third, all animals are multicellular, and most animals have specialized body parts. For example, most animals possess sensory organs like eyes, ears, nose, tongue, antennae, whiskers, etc., enabling them to acquire information about their environment and, among other things, detect potential prey and predators. There are a few other general features of animals. Most reproduce sexually, for example, but these 3 criteria are the easiest to apply when judging whether a particular organism is an animal. And that brings us to our take-home message for section 13.1. All animals eat other organisms, can move during at least one stage of their development, and are multicellular. Moving on, there are 2 possible states for a species. It can be extant, meaning that it currently exists, or it can be extinct, meaning that no living individuals exist. From an evolutionary perspective, referring to any extant species as quote-unquote lower or quote-unquote primitive does not make sense. Rather, these labels usually reflect nothing more than how similar a species is to humans. In this way, we sometimes reveal our biases when speaking of other organisms. Recall that evolution can lead to greater adaptation between an organism and its environment, but it does not necessarily lead to greater complexity or features that can be identified as quote-unquote higher or more advanced. Darwin wrote a note to himself in the margin of a book that proposed a theory of cosmic and biological evolution. Quote, "Never use the word higher or lower," unquote. Throughout his writings, for the many species he described, Darwin emphasized the process of adaptation and the relationship between populations of organisms and the environment in which they lived. One species was never better or worse than another. Rather, each species was differentiated from the others with specializations that adapted individuals of that species to the particular niche in which they lived. The fact that a species exists means it can do the things all organisms must do: find food, escape predators, and reproduce. In this chapter, we investigate 9 of the phyla. There are about 36 animal phyla in all, with the most species and the evolutionary innovations that contributed to their success. These groups represent only 1/4 of all animal phyla, but they account for an estimated 99% of the animal species on Earth today. We begin our discussion of the animal kingdom with animals having a simple body plan, such as sponges, jellyfishes, and worms. We continue through the mollusks and arthropods, complex animals that do not have backbones. And we finish with the vertebrates, mammals, including humans, amphibians, reptiles, including birds, and fishes. Taking this path allows us to explore the major evolutionary transitions in the chronological order in which they occurred. This order does not provide any information about the relative success of any species, nor does it imply that humans are the quote-unquote crown of creation. And that brings us to our take-home message for section 13.2. Of the approximately 36 animal phyla, 9 phyla account for more than 99% of the described animal species. From an evolutionary perspective, no species among these is higher or lower. Moving on, about 2/3 of almost 200 million identified species on Earth are animals. These animals share a common ancestor, a free-living unicellular organism resembling a sperm in size and shape. Analyses of DNA and RNA sequences have helped biologists identify which species share a common ancestor. These analyses have made it possible to better classify the animals into monophyletic groupings. Each of these groupings hinges on a particular adaptation and on the question of whether the animals of a species have descended from an ancestor with the adaptation. Generally, the appropriate placement of species in a phylogenetic tree is based on answers to the following questions. 1. Does the animal have specialized cells that form from defined tissues? Animals having no tissues, that is, groups of cells with a common structure and function, belong to the phylum Porifera, the sponges. Sponges are aggregations of similar cells with little coordination of activities among the cells. All other animals form a monophyletic group with clearly defined tissues. Humans, for example, have highly specialized cells such as skin, muscle, and sensory cells. For all non-sponge animals, we can ask a second question, and that is, does the animal develop with radial symmetry or bilateral symmetry? All animals with defined tissues develop either radial or bilateral symmetry. Radial symmetry describes animals with bodies structured like pies, such as jellyfishes, corals, and sea anemones. Slow-moving or free-floating, radially symmetrical animals have no front or back ends. It is possible to make multiple slices through the center that divide the organism into identical pieces. Other animals demonstrate bilateral symmetry. These organisms, such as humans, cows, and scorpions, have left and right sides that are mirror images. The animals can move adeptly, searching for food and avoiding predators. In some animals, such as starfish, embryonic development is bilaterally symmetrical, but the animal becomes radially symmetrical as an adult. Any animal showing bilateral symmetry at any stage in life is considered bilaterally symmetrical. Among the monophyletic group of animals with defined tissues and bilateral symmetry, there is a further phylogenetic division based on the answer to a third question. And that question is, during gut development, does the mouth or anus form first? Early in the evolution of animals, about 630 million years ago, a major split occurred between protozomes, which can be translated as mouth first, and deuterozomes, which can be translated as mouth second. These names refer to the way the gut develops, from front to back versus from back to front. In protozomes, the gut develops from front to back. The first opening that forms becomes the mouth of the adult animal, and the last opening becomes the anus. In deuterozomes, the gut develops from the back to the front. The anus is the first opening to form, and the mouth is the second. Although it's something you can see only when closely observing animals' embryonic development, the protosome-deuterosome split is one of the most basic divisions of animals in terms of revealing their evolutionary relatedness to one another. In building an overall phylogeny of animals, we can further subdivide into monophyletic groups the animals that have 1. Defined tissues, and 2. Develop with bilateral symmetry, and 3. Are protostomes. To do this, we need to ask a fourth question, and that is, does growth occur by molting or by continuous addition to the animal's body? Animals that molt, such as lobsters and insects, shed their exoskeleton, the hard outer layer, replacing it with the larger one, at intervals during development, and subsequently grow to fit their new exoskeleton. Other animals, such as clams and octopuses, grow by adding to the size of their body in a continuous manner. Moving on, these 4 key distinctions help place the approximately 3 dozen phyla of living animals into monophyletic groups. Within any of the groups, we know that individuals are more closely related to one another than to any individuals outside that group. Besides being organized within monophyletic groups, animals are grouped according to the presence or absence of a backbone. And that brings us to the invertebrates. So invertebrates, including sponges, are animals that do not have a backbone. Vertebrates are animals that do have a backbone. Invertebrates account for more than 95% of all living animal species, although they are not a monophyletic group. In fact, the invertebrates include 8 separate evolutionary lineages with an enormous diversity of body forms, body sizes, habitats, and behaviors. All invertebrates, however, are protostomes, except for one large group whose members are deuterostomes, called echinoderms, including sea stars, sea urchins, and sand dollars. This animal phylum shows the same back-to-front gut development as the vertebrates, and these animals are the vertebrates' closest relatives. We examine them in detail in the following sections. Note that any phylogeny, including the one shown in Figure 13.3 on page 376 of your book, is a hypothesis and, as such, is subject to revision as we accumulate more evidence. Recently, for example, researchers have made the surprising suggestion that perhaps the sponges are not the first lineage of organisms to branch off from the rest of the animals. DNA sequence data from the marine group called ctenophores, previously believed to be most closely related to the cnidarians, indicate that they may have been the first to diverge from the rest of the animals. This idea is surprising because, unlike the sponges, the ctenophores, also called comb jellies, have a nervous system and several types of defined tissues. If the ctenophores were the first animal group to diverge, then the ancestor of all animals would have had a nervous system, which was subsequently lost by the sponges. Alternatively, the nervous system must have evolved independently in the ctenophores. In either case, this proposed new phylogeny may change our understanding of the earliest events in animal evolution. And that brings us to our take-home message for section 13.3. Animals probably originated from an ancestral protist. 4 key distinctions divide extant animals into monophyletic groups. 1. Tissues or not. 2. Radial or bilateral symmetry. 3. Protostome or deuterostome development. 4. Growth through molting or through continuous addition to the body. Moving on, we begin our investigation of animal diversity with the group that may be the least animal-like and the simplest of all animals, the sponges. The sponges belong to the phylum Porifera. As we have discussed, sponges have no specialized tissues or organs, and most lack any symmetry. Despite their simplicity, sponges are remarkably efficient at obtaining food. Living as sessile suspension feeders, sponges strain suspended matter and food particles from water. Sponges have several different cell types. Flattened epithelial cells, skin-like cells connect to each other which cover the outside of the sponge, and collar cells, each of which has a long whip-like flagellum and covers the inside of the sponge. The middle layer is a gel incorporating a diversity of cell types involved in reproduction, digestion, and growth, and it is embedded in the fibers that stiffen the sponge's body. Moving on, the beating of each collar cell's flagellum creates a current that carries water upward and out of the body. An inward flow of water through the pores on the sides of the sponge carries bacteria and algae and other microscopic organisms into the sponge's body. In the quote-unquote collars of the collar cells, sticky mucus traps some of this material, and some particles are taken up into the cells by endocytosis. The volume of water moving through its body daily can be as much as 20,000 times the volume of the sponge itself. Also, sponges are hermaphrodites. That is, each individual contains both male and female reproductive organs, but it produces only one kind of gamete, eggs or sperm at a time. Sponges that are acting as males release a cloud of sperm that swim to other sponges, which are acting as females, and fertilize their eggs. The eggs develop into larvae, which are released into the water and then drift for a few days before settling on a rock or coral outcrop and developing into a sessile adult sponge. Sponges also reproduce asexually. Small buds form on the exterior and eventually break off, settle to the bottom, and may grow into new sponges. Most remarkably, sponge cells are able to reassemble. Picture this experiment. Puree a living sponge in a food blender, then strain the liquid through a fine sieve to remove the chunks, leaving a suspension of individual cells, and then dump that into an aquarium. Within a day, you'll see small clumps of sponge forming as individual cells move around and attach to each other. And within a week, a new sponge will form. You can repeat the experiment with 2 sponge species of different colors. Not only do the sponges reassemble, but the cells from each species assemble only with other cells of the same species, so you'll have 2 sponges, each with only the cells of its own species. Because the dried skeletons of some sponges are soft and absorbent, humans have long made use of them as padding in helmets, as tools for painting, and in ancient Rome in place of toilet paper. But these sponges are far from living. They are just a matrix of soft, elastic fibers of a protein called spongin, isolated from dried sponges. Because overfishing has reduced sponge populations, they are no longer widely available. Note that the sponges used in most kitchens and bathrooms today are nearly always produced from synthetic materials, and loofah sponges, contrary to their name, are made from plant material. And that brings us to our take-home message for section 13.4. Sponges are among the simplest of the animal lineages. A sponge has no tissues or organs. Sponges reproduce sexually by eggs and sperm and asexually by budding. Fertilized eggs grow into free-swimming larvae that settle and develop into sessile, filter-feeding adult sponges. Moving on, if you feel a sting when swimming in the ocean, you may have met a jellyfish. If you are lucky, it will be the mild sting of one of the thousands of species that feed on tiny floating organisms called plankton. Other jellyfish species have powerful stings that can cause extreme pain or even death. The jellyfishes, along with sea anemones and corals, belong to the phylum Cnidaria. Like all animals, except the sponges, the 11,000 species in this phylum have defined tissues. The cnidarians have a simple, radially symmetrical body plan. So, there are 2 types of cnidarian bodies, a sessile polyp and a free-floating medusa. In some species, individuals spend part of their life cycle as a polyp and part as a medusa. Other species exist only as medusa, or as in the corals and sea anemones, only as polyps. Also, cnidarians are carnivores, using their tentacles to capture and feed on marine organisms ranging from protists to fish and shellfish. Their method of capturing prey relies on the stinging cell called a cnidocyte that is unique to the cnidarians. All cnidarians have tentacles located near their mouth that are armed with rows of stinging cnidocytes. Each cnidocyte has a coiled thread with barbs inside and a trigger on the outside. When prey comes into contact with the trigger, the coiled thread is ejected and, like a harpoon, can penetrate the prey, often injecting a toxin. The Portuguese man-o'-war is one of the dangerously poisonous species of cnidarians. Being stung by a Portuguese man-o'-war is painful, and it is extremely painful if the unlucky swimmer becomes entangled in the tentacles and receives hundreds of stings. But these encounters are rarely fatal. We explore here some of the great diversity among the three familiar groups of cnidarians. So that brings us to the corals. Corals live as small, just a few millimeters long polyps in a large colonial group, familiar as the beautiful structures found in coral reefs. Corals sting prey and use their tentacles to catch plankton and occasionally small fish. The prey is directed into the mouth by the tentacles and then digested in the stomach. Corals most commonly reproduce sexually using both external fertilization, that is, releasing sperm and eggs into the water where they fuse and form larvae, and also internal fertilization, in which only sperm are released and can fertilize eggs within female corals. Corals can also reproduce asexually. Corals grow in a variety of shapes, from freestanding, spherical, or branched forms to crusts on rocks or other corals. This is possible because polyps secrete calcium carbonate, creating hard shells on which other corals can live as a colony. Such assemblies of giant calcium carbonate skeletons in corals are called coral reefs. Coral reefs provide an environment that is home to a great diversity of marine life. For instance, the Great Barrier Reef, located in the Coral Sea off the coast of Australia, extends more than 1,500 miles, that's about 2,600 kilometers, from north to south. An awesome display of biological productivity, this reef is the largest biological structure in the world and is visible from the International Space Station. Coral reefs are among the first casualties of global climate change. Coral polyps obtain most of their nutrition from symbiotic algae called zooxanthellae living within the polyps. These algae use carbon dioxide produced by the polyps to photosynthesize, and the polyps gain oxygen and nutrients produced by the algae. Strangely, when coral polyps get too hot, they expel their zooxanthellae. Because zooxanthellae are responsible for the colors of many corals as well as much of their nutrition, when the coral polyps expel their zooxanthellae, the coral appears white, a phenomenon called coral bleaching. The zooxanthellae may return to the polyps when the water cools, but the repeated bleaching events often lead to the death of the coral polyps. The world's oceans are already warming, and coral bleaching events are becoming more frequent. And that brings us to the sea anemones. Sea anemones have polyp bodies that resemble flowers. The anemone has tentacles with stinging cells at the top of its body surrounding its mouth. Sea anemones also have a larval stage that swims freely, then settles on a rock and metamorphosizes into an adult form. Even as adults, many sea anemones can crawl a few inches a day, and wandering sea anemones are often found in seaweed. Jellyfishes range tremendously in size. The Asian giant jellyfish is more than 6 feet across and weighs nearly 500 pounds. Swarms of these giants clog the nets of fishing boats with sticky masses of toxic stingers. At the opposite end of the size scale is the irukandji jellyfish, which is about the size of a hen's egg. This species is so deadly that in 2007, the sighting of just 5 irukandji off the Australian coast halted the filming of a Hollywood movie, Fool's Gold, which had to be completed in the safety of a film studio. And that brings us to our take-home message for section 13.5. Corals, sea anemones, and jellyfish are radially symmetrical animals with defined tissues in the phylum Cnidaria. All cnidarians are carnivores and use specialized stinging cells located in their tentacles to capture prey. Moving on, it's a good thing that most biologists don't run bait shops. If they did, a simple trip to buy worms for fishing would be too complicated. The name quote-unquote worm is commonly applied to a long, skinny, slimy animal without a backbone, but you can find animals fitting that description in 8 different phyla. So worms do not make up a monophyletic group. Their general body plan has evolved several times independently. Before their evolutionary relationships were understood, these groups were classified just as they first were by Linnaeus, as worms, simply because of similarities in their physical appearance. For our discussion, we will consider 3 phyla that illustrate most of the diversity of the animals we call worms. Flatworms, which are from the phylum Platyhelminths, roundworms from the phyla Nematoda, and segmented worms from the phylum Annelida. Keep in mind, however, that from an evolutionary perspective, worm is actually an obsolete and misleading label. We know today that segmented worms are probably more closely related to mollusks, including snails, clams, and octopuses, than they are to roundworms, and roundworms probably are more closely related to arthropods, or insects, than they are to flatworms or segmented worms. The worms have defined tissues and are protosomes. The gut develops from front to back. Unlike the cnidarians, however, the worms, along with the remaining animals discussed in this chapter, develop with a body plan characterized by bilateral symmetry, which adapts them to forward movement. So let's start with the flatworms. Unlike the roundworms and segmented worms, flatworms have no body cavity. That is, the space between the body wall and the digestive tract is not a fluid-filled cavity. Possibly as a consequence, the flatworms have no specialized respiratory or circulatory organs and so have a reduced capacity for diffusion of gases and nutrients. Flatworms grow by adding body mass rather than by molting. These organisms have well-defined head and tail regions with clusters of light-sensitive cells called eye spots at the head end, used for navigation. Most flatworms are hermaphrodites, with each individual producing both male and female gametes, and they engage in both sexual and asexual reproduction. Flatworms include more than 20,000 species and can be parasites, flukes and tapeworms, for instance, or brilliantly colored, free-living aquatic creatures. Many flatworms have a digestive system in which the gut has only one opening, requiring them to consume food and eliminate waste through the same opening. The digestive systems of roundworms and segmented worms, in contrast, have 2 openings. One group of flatworms lacking a digestive system is the tapeworms. These parasitic worms live in their host's gut and absorb nutrients from the host directly through their body wall. All 5,000 species of tapeworms are parasites, with most having a 2-stage life cycle split between 2 different host species. For example, the common tapeworm that infects dogs spends half of its life cycle in fleas. The human pork tapeworm splits its life cycle between pigs and humans. Tapeworms have long, flat bodies made up of repeated segments, each of which is a reproductive unit. Mature tapeworms spread by breaking off segments, which are then shed in the feces of infected hosts. And that brings us to roundworms. The roundworms, also called nematodes, are probably the most abundant animals on Earth. A spoonful of garden soil typically contains several thousand individuals, and some species produce more than 200,000 eggs every day. All roundworms grow by molting. More than 90,000 roundworm species have been identified and named, and there may be 5 times as many species not yet identified. Many soil-dwelling roundworms live in the roots of plants, damaging or even killing the plant. About 15,000 species of roundworms are parasites of other animals, and roundworms are responsible for a large number of human diseases. Most roundworms are transmitted by fecal contamination of soil or food. Tiny parasitic roundworms called filariae are responsible for several devastating tropical diseases. Elephantitis, for example, is a disease in which filariae are transmitted by mosquito bites, and they block the lymph ducts so that fluid accumulates in the limbs or scrotum, causing grotesque swelling. These filariae are found in Africa, South Asia, Pacifica, and the tropical regions of the Americas. And that brings us to segmented worms. The segmented worms number about 13,000 species and are easy to recognize by the grooves running around the body that mark the divisions between the segments. The segmented worms are protostomes with defined tissues, and they do not molt. These worms are organized into 3 groups: marine polychaetes, terrestrial oligochaetes, and leeches. Polychaetes are marine worms living on the seafloor. The word polychaete means many bristles, and their combination of segments and bristles makes polychaetes easy to recognize. Some species burrow through the mud and extract organic material from it. Tube worms use sand grains or limestone to make a tube in which they live with just their waving tentacles exposed. Small particles of food trapped in mucus on the tentacles are then transported to the worm's mouth. Earthworms, which belong to the oligochaetes, meaning few bristles, are the segmented worms you are most likely to have seen. The nightcrawler, a typical earthworm, gets its name from its habit of emerging from its burrows on rainy nights to crawl across the surface of the ground. More than 4,000 earthworm species have been named, ranging in size from less than half an inch to the giant Gippsland earthworm, whose length spans 7 to 10 feet. Earthworms consume particles of soil and organic material. The organic material is digested as it passes through the worm's one-way gut, and fecal plus inorganic soil material is excreted as feces called castings. Gardeners value earthworm castings as soil supplements, and earthworms can produce their own weight in castings every day. Earthworms' activities mix the soil components, create a more uniform mixture of nutrients, and expedite the breakdown of organic materials in the soil, making the nutrients available for plants. Because they perform these tasks, earthworms have enormous economic value to agriculture. A study published in 2008 estimated that the economic value of earthworms in Ireland alone to be more than $1 billion per year. Have you ever waded in a pond or swamp and emerged to find a long, dark brown worm clinging to your leg? If so, you have met the third major group of segmented worms, the leeches. Probably you pulled the leech off, not easy because leeches are both slippery and stretchy, and threw it as far as you could. If you had looked carefully, though, you'd have noticed that, like earthworms, leeches have segmented bodies. Not all leeches are bloodsuckers. In fact, more than half of the leech species are predators. For example, the horse leech, which reaches a length of 8 inches, feeds on smaller segmented worms, snails, and aquatic insect larvae. And that brings us to our take-home message for section 13.6. Worms are not a monophyletic group, although all are bilaterally symmetrical protostomes with defined tissues. Moving on, related organisms don't always look similar. Consider the colossal predatory squid, more than 40 feet long, with eyes the size of beach balls and a razor-sharp beak. Then take a small snail, escargot in French, one of more than 700 million consumed as a delicacy in France each year, and an oyster with its nearly 2,000-year-old reputation as an aphrodisiac, which, by the way, is unsupported by any scientific proof. All of these are mollusks, the second most diverse group of animals. More than 100,000 mollusk species have been identified, named, and many more have yet to be named. The members of this large phylum, Mollusca, live in the ocean, in freshwater, and on land, and they include clams, scallops, mussels, and octopuses. They are so diverse that it is difficult to describe any single defining characteristic. The position of mollusks within the animal phylogeny, however, reflects several important characteristics that they all share: defined tissues, bilateral symmetry, and protostome development. Further, all mollusks grow by adding tissue rather than by molting. Several additional features are common to many mollusks. Some groups of mollusks have a shell that protects the soft body, a mantle, the tissue that secretes calcium carbonate to form the shell, and a sandpaper-like tongue structure called a radula that is used during feeding. Here we will examine three of the major groups of mollusks: gastropods, bivalve mollusks, and cephalopods. Animals in these groups share most of the features common to mollusks, but with very different body plans. So first we'll talk about the gastropods. Snails and slugs are gastropod mollusks. Gastropod means bellyfoot, and these mollusks get their name from the expanded foot on the bottom of their body, which allows them to climb a vertical surface and glide across a horizontal one. Snails have a one-piece curled shell, and slugs have just a tiny remnant of a shell covered by the mantle. Found in both aquatic and terrestrial environments, snails and slugs account for 3/4 of all mollusks. The snail's shell serves as its primary protection against predators, but terrestrial slugs and sea slugs, which have very little shell material, need other defense mechanisms. For example, when a terrestrial slug is attacked, it secretes a sticky slime that deters the predator. Some species of sea slugs synthesize toxic chemicals, while those feeding on sponges can recycle sponge toxins into their own slime. Sea slugs that consume sea anemones can transfer the anemone's stinging cells to the sea slug's own skin, enabling it to sting other creatures. And that brings us to the bivalve mollusks. The bivalve mollusks are soft-bodied animals protected by a pair of shells hinged together by a ligament. Clams, scallops, oysters, and mussels are examples of bivalves. Most of the approximately 8,000 bivalve species live in the ocean, although there are some freshwater species. Clams spend their lives buried in mud or sand, scallops live on the seafloor, and oysters and mussels fasten themselves to underwater objects such as rocks, the pilings that support piers, and the hulls of boats. All bivalves are filter feeders that draw a current of water in through a tube called an in-current siphon and across their gills where tiny food particles are captured. The water then exits through the excurrent siphon. When a grain of sand is trapped in the mantle tissue of a bivalve or between the mantle and the shell, the mantle may secrete layer after layer of shell-like protein material that covers the sand grain and forms an iridescent gem called a pearl. Oysters are the best-known source of pearls, but clams and other bivalve mollusks can also form pearls. And that brings us to the cephalopods. The third major group of mollusks, the cephalopods, includes 6 nautilus species and more than 600 species of squids and octopuses. The nautilus has an external shell. Squids have very small shells that are covered by the mantle, and octopuses have lost the shell entirely. In these species, tentacles appear to grow directly from the head, which explains the name cephalopod. Cephalic means of the head, podia means of the feet, therefore cephalopod translates as head-footed. Cephalopods use their tentacles to walk, swim, and capture prey. Squids, many of which are ferocious predators, have 8 short tentacles called arms and 2 long sucker-bearing tentacles used to capture prey. A squid can propel its tentacles forward with astonishing speed. Accelerations of more than 800 feet per second have been measured, the equivalent of your car accelerating from 0 to 6,000 miles per hour in 10 seconds. The suckers on the tentacles adhere to the prey and draw it back toward the squid. Many squids are more than 3 feet long, and members of some species may exceed 40 feet. When a squid is in a hurry, it swims tail end first using jet propulsion. Water is expelled through the siphon at the tentacle end of the body, shooting the animal backward. Octopuses, which have tentacles that spread 12 feet, are bottom dwellers that live among coral reefs and on rocky coasts. They can squeeze through astonishing small openings, and their ability to escape from sealed tanks is a perpetual challenge for the keepers of zoos and aquaria, who are accustomed to arriving in the morning to find an octopus wandering around the room. So, octopuses are very good at manipulating things with their tentacles. It doesn't take a captive octopus long to learn how to unscrew tops on glass jars to reach fish swimming inside. That feat is a minor variation on twisting a clam to open it for food. For this reason, octopuses are sometimes featured in online videos and TV shows and described as the smartest invertebrates, though this is the sort of claim we must view skeptically. What is intelligence? The concept of intelligence loses its meaning when we are comparing animals that live in completely different environments and respond to entirely different stimuli. We can say that octopuses are very good at doing things that octopuses need to do, searching for prey, capturing it, and manipulating it, for example. But is following a complex sequence of actions the same thing as intelligence? Spiders, for example, construct elaborate webs that are strategically placed in the flight paths of insects. They detect the impact of an insect on the web and rush out to wrap their prey in silk before it can escape. Does that require more or less intelligence than opening a glass jar to eat a fish? And what about the red squirrels of northern Europe? Every fall, a squirrel can hide more than 3,000 acorns, cones, and nuts. Over the course of the winter, it may then recover more than 80% of these hidden nuggets of food. If intelligence is defined in part as the ability to do some problem-solving task valued by humans, the concept of quote-unquote intelligence may be relevant only to humans. For this reason, questions of comparative animal intelligence are not generally useful. And that brings us to our take-home message for section 13.7. Mollusks are protostome invertebrates that do not molt. They are the second most diverse phylum of animals and include snails, slugs, clams, oysters, squids, and octopuses. Moving on, when it comes to species diversity, the arthropods take the cake, outnumbering all other forms of life. This phylum, Arthropoda, contains almost 1 million recognized species, or approximately 75% of all animal species and 60% of all species on Earth. And about 80% of arthropods are insects. There are approximately 6 times as many species of beetles as species of all birds, mammals, fishes, amphibians, and reptiles combined. Not only are the arthropods diverse, but they're also extremely abundant. Taken together, there are more than 1 billion, that is a 1 followed by 18 zeros, arthropods alive at any given time. Put another way, right now there are more than 150 million insects for every single human. So, the Arthropoda phylum contains bilaterally symmetrical protostome invertebrates that have distinct body segments, a rigid external covering called an exoskeleton, which is made of a stiff carbohydrate known as chitin, and legs with joints. Think of all the joints in a lobster's claw. The four major lineages of arthropods are millipedes and centipedes, chelicerates, including horseshoe crabs, spiders, mites, ticks, and scorpions, crustaceans, including lobsters, crabs, shrimps, and barnacles, and insects. The arthropod phylum is so large that even if we excluded all the insects, the most diverse arthropod group, it would still contain more species than any other phylum. So let's first talk about millipedes and centipedes. Millipede and centipede translate into 1,000 feet and 100 feet, respectively, and refer to the enormous number of legs on these long, skinny animals. They don't really have 1,000 or even 100 feet, but millipedes do have more legs than centipedes, and that is the easiest way to tell them apart. Millipedes and centipedes have long, segmented bodies that seem almost worm-like, but their jointed legs and hard exoskeletons are characteristics of arthropods. Both live among fallen leaves, especially in forests where the leaf litter layer is cool and moist. Millipedes feed on decaying plant material. Centipedes, however, are predators that use fangs equipped with venom to kill insects and even snakes and small mammals. The centipedes then use their jaws to tear their prey into pieces small enough to swallow. And that brings us to the chelicerates. There are about 60,000 species of chelicerates, a group that includes 4 horseshoe crab species, and the arachnids, land-dwelling arthropods that include spiders, scorpions, mites, and ticks. Most have 4 pairs of walking legs and a specialized feeding apparatus. Arachnids are predators. Many spiders construct webs by spinning silk fibers consisting of interconnected protein molecules. Orb-weaving spiders construct intricate spiral-shaped webs that trap and sometimes even lure flying insects. When a spider captures prey, it uses fangs to inject its victim with venom containing 2 types of enzymes. The first set of enzymes disrupts the prey's nervous system and paralyzes it. The other enzymes then dissolve its internal organs. The spider sucks out the liquefied contents. The venom of most spiders is usually harmless to animals as large as humans, but 2 North American spiders do pose a risk to people. The black widow spider is identified by a red hourglass marking on the bottom surface of the female. The brown recluse spider lacks distinctive markings, so it is harder to identify and more dangerous. Both species often move indoors, constructing webs in closets and behind furniture. Brown recluse bites create wounds that remain open for months and sometimes required repeated skin grafts to close. Scorpions look dangerous to humans, yet the sting of North American scorpions is no worse than a bee's. The sting of the African scorpions, however, can be lethal to humans. Scorpions are deadly predators for insects, though. They seize insects with their claws and use their stinger only if the prey struggles. Some of the scorpion's mouth structures are covered with spines that grind the prey, while glands secrete enzymes that liquefy it, and the scorpion sucks up the soup. And that brings us to crustaceans. In the U.S., the top 3 fishery crops by annual market value are crustaceans: shrimp, crabs, and lobsters. There are about 52,000 species of crustaceans. Although these species can look very different, ranging from sessile barnacles to mobile tiny krill, they all share one feature in common: 5 pairs of appendages extending from the head. 3 of these appendages are used for feeding, and 2 are antennae that sense the environment. Crustaceans have many pairs of legs, which can be modified for many purposes. Shrimp and barnacles. Barnacles are closely related to shrimp but spend their adult lives within a shell and fastened to a rock, have legs with comb-like projections that they use to capture plankton. Free-living crustaceans such as shrimp, crabs, lobster, and crayfish have modified limbs in the abdominal region used to hold eggs or newly hatched young. Although most crustaceans are aquatic, the inconspicuous woodlice, also known as pillbugs, sowbugs, or roly-polies, are terrestrial forms that you can find when you turn over a rock in your garden. Woodlice are herbivores and play an important role in recycling dead plant material. And that brings us to insects. Mostly terrestrial, insects have 3 pairs of walking legs, and most also have 1 or 2 pairs of wings, extending from the midsection. Insects abound in nearly all terrestrial habitats and are the most diverse group of arthropods. Every species of plant, for example, is eaten by at least one insect species. Wings and the ability to fly, as well as the developmental process of metamorphosis, have played an important role in the great success and diversification of insects. And that brings us to our take-home message for section 13.8. The arthropods are protostome invertebrates. They outnumber all other forms of life and species diversity. Insects are the most diverse group of arthropods. Moving on, 2 characteristics of insects that have been central to their success are the ability to fly and the way they cope with change in body size as they grow. Wings are adaptations that first appeared in insects. With flight, insects can avoid many predators and can efficiently search for food and for mates. Flight also enables organisms to more quickly leave one location and disperse to another that is potentially more hospitable. As arthropods, insects have a rugged exoskeleton to which their muscles are attached. Like a suit of armor, this exoskeleton provides excellent protection from injury and predators. It also helps individuals conserve water and resist drying out. The exoskeleton, however, prohibits growth. To overcome this constraint, all arthropods, as well as the roundworms of the phylum Nematoda, rely on a process of growth that includes molting, which is very different from the growth pattern in other animals. So, after hatching, their life is divided into 3 completely different stages. First is larva. An egg hatches into a larva, which looks completely different from the adult form. A caterpillar is the larval stage of a butterfly or moth, and a grub is the larval stage of a beetle. A larva's job is to eat and grow large enough to enter the next stage of life, the pupa. Larvae grow by passing through numerous stages, usually 4 to 8 stages, in which they shed their exoskeleton, increase in size, and develop a new, slightly larger exoskeleton in the process of molting. Second is pupa. Eventually, a larva completely covers itself with a casing, such as a cocoon or a chrysalis, at which point the insect is called a pupa. In the pupa, genes are activated that code for the adult body form, and the proteins that made up the larva are broken down into amino acids, which are recycled to synthesize adult proteins and reassembled into the adult form. This rebuilding process is called metamorphosis, from 2 Greek words meaning change of form. The entire process requires several days, and by the end of the pupal stage, the adult insect is curled tightly inside the pupal case. And third is the adult. When the adult form hatches from the pupal case, it is at its full size. For this reason, the adults of some insects do not even eat. Rather than eating and growing, the job of an adult insect is reproduction. So the developmental process called complete metamorphosis, which includes the 3 stages, occurs only in insects and allows the larva and adult to act as if they were animals from different species, each optimized to perform very specialized tasks. Caterpillars eat leaves, for example, and spend their entire larval period on a single plant. In contrast, butterflies feed on nectar that they collect by flying from flower to flower, saving up enough resources to lay or fertilize eggs. As a larva, the animal feeds and grows. As an adult, it reproduces. The genes that control the larval body form are different from the genes that determine the adult form, so natural selection can act on the larval and adult stages independently. About 83% of insect species go through complete metamorphosis, and this separation of the life stages has been an important factor in helping insects diversify. There are nearly 20 times as many named species of insects as there are named vertebrate species. Among those species that do not undergo complete metamorphosis, typically the eggs hatch as nymphs, the juvenile form, which resemble a smaller version of the adult. These juveniles do not have wings or reproductive organs, but they live in the same habitats as the adults and eat the same foods. They then undergo several molts as they grow and stop molting when they reach adult size. This pattern of growth and development, called incomplete metamorphosis, occurs in grasshoppers and cockroaches, among many other insect species. And that brings us to our take-home message for section 13.10, the ability to fly and the developmental process of metamorphosis as a means of overcoming the constraints imposed by a rugged exoskeleton have contributed to the enormous ecological diversity of insects. Moving on, appearances can be deceiving. Sea stars, sand dollars, and sea urchins are members of the phylum Echinodermata and live in the oceans. They are radially symmetrical as adults and are covered by spiny plates. And yet, because they are deuterostomes, in other words, their gut forms from back to front, they are actually more closely related to humans than to any other vertebrate group. This unexpected relationship reveals the force of natural selection and the dramatic effects it can have on body form as populations of organisms become adapted to their environment. So, the ectoderms include about 6,000 species of marine animals, most of which are enclosed by a hard skeleton of spiny plates. Adult sea stars, starfishes and brittle stars, probably the most recognizable echinoderms, have 5 or more appendages evenly distributed around their body circumference. They move with equal ease in any direction, and their sensory organs are distributed around their circumference. Adult sea urchins and sand dollars don't have projecting arms, but are also radially symmetrical. Although radial symmetry is a characteristic of adult echinoderms, their larvae are bilaterally symmetrical. The structure of the larvae provides evidence that echinoderms evolved from bilaterally symmetrical ancestors, and their radial symmetry as adults is an adaptation associated with their mode of locomotion and feeding specializations. Echinoderms do not have a brain. Instead, they have a nervous system that consists of a central ring of nerves with branches that extend into each of their appendages and help them gain information about and respond to their environment. Echinoderms creep on little tube feet that are extensions of an internal system of water-filled canals radiating throughout their body. Tube feet carpet the undersides of sea stars' arms and extend up and around the bodies of sea urchins and sand dollars, extending and contracting in waves to grasp and release the surface on which they move as the animal glides along. Although each tube foot is tiny, there are thousands of them, and their combined force allows a sea star to pull apart the 2 shells of a clam or mussel. Once the shells are pried apart, the sea star pushes its stomach out through its mouth and inserts it into the open shells. The stomach then secretes digestive enzymes that break down the clam or mussel tissue, and the sea star absorbs the soup that results. Sea urchins feed on algae, scraping them loose from rocks with sharp tooth-like surfaces made from calcium. Sand dollars capture floating particles of algae and other organic matter by trapping them in streams of mucus, which are moved toward their mouth by beating cilia. And that brings us to our take-home message for section 13.11. Because they are deuterostomes, as are vertebrates, echinoderms are the invertebrates that are the closest evolutionary relatives to the vertebrates. Most, including sea stars, are enclosed by a hard skeleton of spiny plates and are radially symmetrical as adults. Moving on, vertebrates are deuterostome animals having defined tissues and bilateral symmetry. They are part of the phylum called Chordata, the chordates, which includes three major groups, the vertebrates and two invertebrate groups, the tunicates and the lanceolets. So, members of all three of the major groups of the chordates possess, for at least a portion of their life cycle, four distinctive chordate body structures that are found in no other animal groups. First, the notochord, a rod of tissue extending from head to tail. This is the structure that gives chordates their name. The notochord, which is replaced by the backbone in vertebrates as the embryo develops, stiffens the body when muscles contract during movement. Second is a dorsal hollow nerve cord that extends along the animal's back from its head to its tail. In vertebrates, this nerve cord eventually forms the spinal cord and the brain. Third are gill slits, which are present in the embryos of all chordates, but in many chordates, including humans, the slits disappear as the animal develops. And fourth is a post-anal tail, which is another chordate characteristic. All vertebrates have a tail in this location, but some, including humans, have only a tail for a brief period during embryonic development. So tunicates, Eurochordate of about 2,000 species, are invertebrate marine animals that have defined tissues, bilateral symmetry, and deuterostome development in which their gut develops from back to front. The adults are thumb-sized and look like balls of brownish-green jellyfish. Lanceolets, Cephalochordata, about 20 species are known, are also bilaterally symmetrical, filter-feeding deuterostomes. These slender eel-like invertebrate animals have the length of about your little finger and live in coastal waters. And then there's the vertebrates, which number about 56,000 species, and these are the most diverse group of chordates. Because they are deuterostomes, they are bilaterally symmetrical with defined tissue and back to front gut development. But vertebrates differ from the other chordates in two important ways. First, they have a backbone, formed when a column made from hollow bones or cartilage in some species, called vertebrae, forms around the notochord. This backbone surrounds and protects the dorsal hollow nerve cord. Second, they have a head at the front or the anterior portion of the organism, and the head contains a skull, a brain, and sensory organs. So the earliest vertebrates were fish-like animals that lived more than 500 million years ago and did not have jaws. Today, just two kinds of jawless vertebrates exist, the lampreys, which number about 41 species, and the hagfishes, which number about 43 species. Fishes swim in the same way that lampreys and hagfishes swim, that is, by bending the body from side to side to create an S-shaped wave that moves from head to tail. Unlike the simple tube-like, limbless, and finless body of hagfish and lamprey, a typical fish has 7 fins serving different purposes: to drive the fish forward, to minimize rolling from side to side, and for steering and stopping. The combined effects of the 7 fins allow the fish to swim rapidly in a straight line in open water, or to weave its way through a dense stand of plants or corals. Fins propel the organism to its prey, and jaws capture and kill it. The evolution of fins and jaws set the stage for the explosion of diversity in vertebrates. So, there are three categories of jawed fish: cartilaginous fishes, ray-finned fishes, and lobed-finned fishes. Cartilaginous fishes, which number about 880 species, include sharks and rays. Cartilaginous fishes are characterized by a skeleton made entirely of cartilage, a solid but slightly flexible connective tissue, the same tissue that gives your nose and ears their shape. Ray-finned fishes have a rigid skeleton made from bone. Like cartilage, bone is a solid connective tissue consisting of specialized cells and an external material, the matrix, that the cells secrete. Bone, however, is much less flexible than cartilage because its extracellular matrix is mineralized by crystals of calcium phosphate. Ray-finned fishes have a mouth at the narrow tip of the body, and their fins made from webs of skin are supported with hardened rays made of bone. Ray-finned fishes are the largest group of jawed fishes and the most diverse group of vertebrates, with about 27,000 species known. This group includes almost everything you think of as a quote-unquote fish, from salmon to goldfish. In addition to the bony skeleton, an important evolutionary development in ray-finned fishes was the swim bladder, a gas-filled organ that keeps the fish from sinking. This bladder is believed to be homologous to the lungs of land-dwelling terrestrial vertebrates, with both evolving from simple air sacs involved in gas exchange connected to the gut. Cartilaginous fishes, which have no swim bladder, must constantly move through the water or they will sink. The third group of jawed fishes, the lobed-finned fishes, are represented by just 8 known species. These fishes have sturdy fins on the underside of their body. Unlike the fins of ray-finned fishes, these lobe fins have a central appendage containing numerous bones and muscles that connect the fins to the body. As we will see in the next section, lobe fins were useful in initiating the move onto land. And that brings us to our take-home message for section 13.12. Chordates, including vertebrates, the most diverse group of chordates, have a notochord, a dorsal hollow nerve cord, pharyngeal slits, and a postanal tail. Among vertebrates, the development of 2 structures in fishes, fins and jaws, set the stage for the enormous diversity of modern vertebrates. Moving on, the ability to live on land brought significant evolutionary benefits, including reduced competitive pressure from all other marine life and a rich panoply of new habitats to utilize. Movement from water onto land, however, required 4 major evolutionary adaptations: lungs, a backbone, 4 legs, and eggs that don't dry out. All 4 characteristics are necessary for a land animal, and all 4 evolved in predatory fishes that lived in shallow water, the immediate ancestors of terrestrial vertebrates. As we've noted, lungs probably evolved from the air sacs that were also evolutionary precursors to the swim bladder found in ray-finned fishes. All of the terrestrial vertebrates, amphibians, reptiles, including birds, and mammals are descendants of lobed-finned fishes that lived during the Devonian period some 400 million years ago. At this time, the lobed-finned fishes had lungs and rudimentary limbs in the form of fins, but they were still fully aquatic and living near the shore. The 4 sturdy fins on the underside of their body helped them move through shallow water, and their lungs allowed them to breathe air when the oxygen concentration in the warm, stagnant water was low. Thus, these fishes already had some of the basic characteristics of terrestrial animals. The jointed bones in the fins of lobed-finned fishes don't look much like your arms and legs, but they are homologous structures. To move onto land, a vertebrate needed more than just legs and lungs. It needed structural support to resist the pull of gravity. Each vertebra of a terrestrial vertebrate has projections that interlock with projections from both the vertebrate ahead of it and the vertebrate behind of it. These interlocking projections prevent the backbone from sagging under the pull of gravity, and body weight is transmitted through the limbs to the ground. The innovation necessary to move onto land was an egg that resists drying out. When eggs are deposited on land, they are exposed to air and lose water by evaporation. The eggs of terrestrial animals need a waterproof protective covering, a membrane, and a shell. The shell appeared about 380 million years ago and further facilitated the evolution of entirely terrestrial vertebrates, the groups that evolved into mammals and reptiles. And remember, reptiles include birds. These initial egg coverings were soft and probably still required a watery environment. Later, the eggs of terrestrial vertebrates acquired a watertight membrane that keeps the embryo surrounded by a bath of fluid called amniotic fluid. And that brings us to our take-home message for section 13.13. In the transition from life in water to land, 4 adaptations were important: lungs, a backbone, 4 legs, and eggs that do not dry out. Moving on, the ancestors of all vertebrates that live on land had lungs and 4 legs. Some modern vertebrates, such as whales and snakes, have evolved so that their limbs are reduced to a few shrunken and unused bones. Traits have been lost in other species too. Some salamanders, for example, have lost lungs and now breathe through their skin. These are recent changes, however, occurring in species whose ancestors had lungs and 4 legs. Taxonomically, all terrestrial vertebrates are tetrapods. Terrestrial vertebrates divided into 2 main groups: non-amniotes, such as amphibians, which reproduce in water and do not have desiccation-proof amniotic eggs, and amniotes, such as reptiles, birds, and mammals, which have amniotic eggs, named for the protective waterproof sac called an amnion that surrounds the embryo. We discuss amphibians here, then cover reptiles and mammals in the next 2 sections. So, the first terrestrial vertebrates were amphibians. Amphibian comes from the Greek word amphibios, meaning living a double life. Most amphibians go through 2 stages: a water-breathing juvenile form followed by an air-breathing adult form. The adults of many amphibian species lay their eggs in water. Amphibian eggs lack the waterproof layers that allow amniotes to lay eggs on land, so most of the 6,000 species in this group are still significantly tied to life in water. Frogs and toads make up the vast majority of amphibians, about 5,400 species. Other amphibians include salamanders, about 550 species, and caecilians, a group of legless burrowing animals, encompass about 170 species. The juvenile stage of a frog, called a tadpole, lives in water, lacks legs, and eats algae. A few weeks or up to many months in some species after hatching, metamorphosis occurs and the tadpole develops legs, lungs, and a digestive system fit for its adult life as a carnivore. Adult frogs have thin, moist skin through which gas, oxygen and carbon dioxide, exchange with the air can take place. When a frog is underwater, oxygen diffuses directly from the water into the frog's blood. The past 2 decades have seen a stunning decline in amphibian populations throughout the world, and almost 1/3 of all amphibian species are endangered or threatened. This situation is attributable to a combination of causes: climate change, habitat degradation, fungal diseases, and increased pollution. Amphibian species are particularly vulnerable to extinction because their very specific microhabitat requirements with respect to moisture, temperature, and pH are commonly disrupted during even minor habitat changes. And that brings us to our take-home message for section 13.14. Amphibians are terrestrial vertebrates, but the adults of most species still lay eggs in water. The eggs hatch into aquatic juveniles. Moving on, soon after amniotic vertebrates appeared, two different evolutionary lineages began to diverge. One of these lineages contains the mammals, the group to which humans belong. The other lineage is the one referred to in this chapter as reptiles, which includes birds. That is an awkward term, but the evolutionary tree shows why it is necessary. Surprisingly, birds, which number about 9,700 known species, are one branch of the reptile lineage that also includes snakes and lizards, and snakes and lizards number about 8,000 known species, turtles, which number about 300 known species, crocodiles and alligators, about 23 species, two species of the New Zealand tuatara, and the dinosaurs. The characteristics shared among the members of the bird-crocodile-dinosaur group are mostly similarities in bones, especially the bones of the skull and legs, and in DNA sequences. Reptiles are amniotes, so they have an amniotic egg. Their offspring develop within a fluid-filled and waterproof sac inside the eggshell. You are familiar with hard-shelled eggs of birds, and some lizards, snakes, and turtle eggs also have hard shells. Other species in these groups have eggs with a paper-like shell. So, most people find it confusing that birds are grouped with turtles, lizards and snakes, and alligators and crocodiles. And that confusion is easy to understand. After all, birds are covered by feathers and can fly, whereas the other extant reptiles have bare skin and do not fly. And there is an important physiological difference as well. Birds are what we call endotherms, meaning they use the heat produced by cellular respiration to raise their body temperature above air temperature, whereas other reptiles are what we call ectotherms, and they heat themselves in the sun to raise their body temperature and seek shade when the air is too warm. So, birds are, in fact, just one group of dinosaurs. During the Mesozoic era, from about 250 million to 65 million years ago, dinosaurs were the dominant terrestrial vertebrates. The fossil record shows a remarkably clear series of animal forms that bridged a transition between bare-skinned dinosaurs and feathered birds. The most readily apparent difference between reptiles, including most dinosaurs, and birds is the presence of feathers in birds. The fossil record reveals that feathers evolved before birds. Fossil evidence also suggests that the initial evolution of feathers probably had nothing to do with flight. Among reptiles, many different species had feathers. For example, a small, agile dinosaur called Sinosopteryx, which lived about 120 million years ago, had feathers that were simple, spiky filaments on its neck, back, and tail, along with shorter filaments covering its body. The 2010 discovery of fossilized pigment-packed organelles called melanosomes within these feathers indicates the feathers and filaments were probably brightly colored. Researchers believe that they may have been used by male Sinosopteryx for courtship displays to females, as well as for aggressive displays to other males. Sinosopteryx and many other feathered dinosaurs were latecomers. The pigeon-sized Archaeopteryx was among the first bird species and was already using its feathers to fly by some 147 million years ago. By 140 million years ago, the skies were filled with birds. Another big difference between birds and reptiles is that, as noted earlier, birds use internally generated heat to maintain a high body temperature, whereas ectotherms rely on the sun to heat their bodies. This evolutionary change may have been related to feathers. As feathers evolved for display and flight, they may also have provided some insulation. This insulation may then have enabled the evolution of high rates of cellular respiration and the maintenance of a high and constant body temperature. And that brings us to our take-home message for section 13.15. Birds are a branch of the reptile lineage, but unlike other extant reptiles, they possess feathers and can generate body heat. Feathers were originally colorful structures, possibly used for behavioral displays. Additional functions such as insulation and flight evolved later. Moving on, as we saw in the previous section, two different evolutionary lineages began to diverge after amniotic vertebrates appeared. One gave rise to reptiles, and the other led to mammals. Originally small nocturnal insect eaters, the early mammals remained small, and judging from the scarce fossil record, they were never in great abundance while the dinosaurs roamed the earth. Early mammals had several features in common that are still present in mammals today. One important feature is hair. Dead cells filled with the protein keratin, which serves as an insulator. Another is mammary glands in female mammals, which enable them to produce nutritious, calorie-rich milk and nurse their young. The evolution of the earliest mammals was marked by a gradual transition from bodies with short legs that projected out horizontally from the trunk, like the legs of an alligator, to bodies with long legs held vertically beneath the trunk, like those of a dog. These anatomical changes would have allowed the earliest mammals to run faster and farther to capture prey, but an increase in muscle activity for running had to be accompanied by an increase in the rate of cellular respiration to supply energy for those activities. With the increase in metabolism, along with the evolution of hair, the early mammals could trap the heat produced and use it for temperature regulation, setting the stage for endothermy, but through a different sequence of events than in birds. So, one common feature of most mammals is viviparity, or giving birth to babies, or we could also call that live birth, rather than laying eggs. But it is not a defining mammalian characteristic. Monotremes retain the ancestral condition of laying eggs. Monotremes do produce milk, but they do not have nipples. Their mammary glands open up directly in the skin so babies can lap up milk from the skin and hair. Today, only 5 species of monotremes survive: the platypus and 4 species of spiny animals called echidnas. The platypus lives in streams and rivers in the eastern part of Australia. It uses its broad, leathery, electrosensitive bill to find aquatic insect larvae and crayfish, which the platypus locates by sensing the electrical activity of its prey's muscles as they try to hide. The echidnas, in which 3 species live in Australia and 1 in New Guinea, also have a leathery, electrosensitive snout. The remaining 2 lineages of mammals, the marsupials and the placentals, are viviparous, but the newborn of young marsupials and placentals are quite different. Marsupials are called pouched mammals because the female of most marsupial species has a pouch on her abdomen in which the young complete their development, following a short period of embryonic life in the uterus. As you might imagine, placental mammals, including humans, take their name from the placenta, the structure responsible for the transfer of nutrients, respiratory gases, and metabolic waste products between the mother and the developing fetus. The placental fetus has a much longer period of development in the uterus than does the marsupial fetus. And that brings us to our take-home message for section 13.16. Hair and mammary glands are defining characteristics of mammals. Moving on, the primates, the evolutionary lineage to which humans belong, originated about 55 million years ago. We can get an idea of what the ancestral primate looked like from the modern species of tree-living arboreal mammals, commonly called prosimians, a group that includes the lemurs. This is because many of the anatomical characteristics of humans and other primates can be traced to our arboreal origin. Our forward-directed eyes and binocular vision that allow us to judge distances accurately, our shoulder and elbow joints that allow our arms to rotate, and our fingers and opposable thumbs that allow us to grasp objects. All these traits we inherited from our arboreal ancestors. So humans are part of the primate lineage that includes the groups commonly referred to as the New World and Old World monkeys, both of which have tails, and the apes, which lack tails. Within the apes, genetic and anatomical characteristics show that chimpanzees are our closest living relatives. Human and chimpanzee genes are very similar. Their base sequences differ by only about 1%, and 1/3 of human and chimpanzee genes are identical. The amount of genetic difference between humans and chimpanzees indicates that the chimpanzee and modern human lineages separated only 5 or 6 million years ago. Humans differ from chimpanzees in 3 major anatomical characteristics. Humans are bipedal, in other words, we normally walk on 2 legs, whereas chimpanzees usually walk on 4 legs. Humans are bigger than chimpanzees, and the human brain is about 3 times the size of the chimpanzee brain. When we trace the appearance of these human characteristics through fossil records, we find that humans did not become bipedal, big and brainy all at once. Bipedality evolved first, then brain volume increased, and finally body size increased, accompanied by a further increase in brain size. Shifting from walking on 4 legs to walking on 2 legs required changes in several parts of the skeleton. The evolution of bipedalism was not a simple process, but it was one of the first important changes in the human lineage, and it seems to have set the stage for all the changes that followed. The primary advantage of bipedal locomotion for early humans was probably energy efficiency, though this hypothesis is still debated as research continues. Bipedal locomotion at walking speed uses less energy than quadrupedal locomotion, and it also frees the hands for carrying and for tool use. So, about 3.5 or 4 million years ago, several bipedal groups known as Australopithecines appeared. They were no larger than the chimpanzees and had the same brain volume, just a bit larger than a pint jar. The fossil known as Lucy was the first of many Australopithecines to be discovered. An adult female, Lucy was only 3 feet tall and probably weighed no more than 60 pounds. The group of Australopithecines to which Lucy belonged lived in a grassy habitat with scattered trees. Their hands and feet retained the curved finger and toe bones that are characteristic of arboreal animals. Lucy and other members of the closely related species of Australopithecines probably foraged on the ground for food and climbed into trees to escape predators and perhaps at night to sleep. With jaws and teeth much like the jaws and teeth of chimpanzees, they consumed a diet probably much like that of chimpanzees, a mixture of leaves, soft fruits, and nuts. Following the appearance of bipedal Australopithecines, a second branching episode in human evolution produced the earliest species in our genus, Homo, the word for human. These earliest humans had brain volumes about twice those of the chimpanzees, but little increase in body size. The species in this radiation had markedly smaller teeth than their Australopithecine ancestors, a change that might indicate they had started to use tools instead of their teeth for the initial preparation of food. Stone tools are found in many of the same deposits as the fossils of human habilis, and the species may have been the first to use tools. Up to this time, all of human evolution had taken place in the southern and eastern parts of Africa. However, studies suggest that about 2 million years ago, one branch, represented by Homo erectus, left Africa and spread through Europe and Asia, while a second branch, represented by Homo ergaster, remained in Africa. Additional branching episodes in human evolution gave rise to several species of humans, including Neanderthals, Homo neanderthalis, Flores man, Homo floresiensis, and our own species, Homo sapiens. This radiation coincided with an increase in body size to approximately the height and weight and body form of modern humans, as well as an increase in brain volume to nearly twice that of our early ancestors. And that brings us to our take-home message for section 13.17. Humans' forward-looking eyes, grasping fingers, and shoulder and elbow joints that allow arms to rotate are characteristics retained from our arboreal ancestors. The early ancestors of humans left the trees and took up life on the ground where they walked on two legs. Moving on, about 200,000 years ago, a new human species branched off of the Homo ergaster lineage in Africa. This was the first modern Homo sapiens. Then, about 60,000 years ago, a small group, probably no more of 100 individuals of these modern humans, left Africa and ultimately spread across the earth. Mitochondrial DNA analyses show that the initial human migration out of Africa followed three major pathways. One path turned west and spread into Europe. A second path turned southeast and spread into southern Asia and through the Indo-Australian archipelago to Australia. The third migration went northeast, populating northern Asia. This group crossed the Bering Strait land bridge about 15,000 years ago and spread southward through the Americas. Even as some groups migrated from Africa, many individuals remained in their original homeland. So there was something about the world that those Homo sapiens groups encountered as they left Africa that we would find bizarre. They were not alone. Populations of at least three other species of humans, i.e. the species of the genus Homo, were present in some of the areas into which the modern humans were moving into. How strange and different that world must have been with multiple human species. What would it be like to live with species that were as similar to us as dogs are to wolves, coyotes, and jackals? How would you interact with an individual who was clearly human, but not human in exactly the same way that you were? This is a situation that the first modern Homo sapiens encountered when they left Africa. Neanderthals had already spread across Europe and the Middle East. Neanderthals were about the same size as modern humans, but more robust and muscular. Fossils of Neanderthals often include bones that had been broken and healed. These injuries provide two types of information about Neanderthals. One, Neanderthals must have lived in organized groups that included a social support system because these fossils reveal serious injuries. The injured individuals would have been incapacitated for days or even weeks until they healed, and family or clan members must have cared for them. Ritualistic burials of Neanderthals have also been found, providing additional support for the hypothesis that these humans lived in organized social groups. Second, the injuries found in Neanderthal skeletons closely resembled the injuries of professional rodeo bull and bronco riders who are trampled by large, angry animals. Neanderthals probably hunted large mammals, bisons, mammoths, woolly rhinoceroses with short spears that were used for close-up jabbing. So, the Homo sapiens who migrated through the Indo-Australian archipelago encountered two species of humans, Homo erectus and Homo floresiensis. Homo erectus had migrated to Asia at least 1 million years earlier and was still present on the island of Java when modern humans arrived about 50,000 years ago. Although Homo erectus was the same size as modern humans, it had a smaller brain, an average brain volume of just over a quart. For Homo erectus, compared with a quart and a half for Homo sapiens, it is hypothesized that Homo erectus may have been the first humans to use fire and the first humans to build boats. Then in 2003, researchers discovered stone tools and fossils of a species of human only 3 feet tall and with a brain volume of just over a pint on Flores Island. Flores Island lies just east of Java. The paleontologist who discovered the species gave it the scientific name Homo floresiensis, Flores man, but the press would promptly call it the Hobbit because of its tiny size. When the dwarf human floresiensis lived, Flores Island was also home to the dwarf elephant, which Homo floresiensis may have eaten, and a giant monitor-sized liver, which may have eaten the floresiensis. So after modern Homo sapiens groups spread into the areas occupied by the three other human species, these three other species disappeared. Neanderthals became extinct about 30,000 years ago. Homo erectus became extinct about 27,000 years ago, and Homo floresiensis about 12,000 years ago. Researchers once thought that modern Homo sapiens exterminated the other species either by monopolizing access to food and living space or by killing them in battles for these resources. But recent results from DNA sequencing suggest that just as they emerged from Africa, Homo sapiens interbred with Neanderthals. This conclusion is based on the finding that less than 1% to 4% of the genetic makeup of most modern humans includes Neanderthal DNA. Now we are only extant humans, a situation that is unique in the evolutionary history of humans. Ever since the first branching event about 4 million years ago, multiple closely related species of humans had coexisted. We are the first human species to live on Earth without related species of humans. And that brings us to our take-home message for section 13.18. Modern humans evolved in Africa about 200,000 years ago. About 60,000 years ago, a small group of modern humans moved out of Africa, and the descendants of this group ultimately populated Europe, Asia, and the Americas.